During this week's lab, you will be completing two activities in which you should use sterile technique: the DNA transformation activity and setting up your plant hormone research project plates. For both of these activities, it is important that you do everything possible to reduce the risk of contamination of your plates so that you will have solid results at the conclusion of these experiments. Accidental introduction of bacteria or fungi can result in the growth of a contaminant that can overtake your organism of study and result in the loss of data for you at the conclusion of your experiment. As a result, you will need to do everything possible when you set up your plates to prevent contamination. We have provided sterile shields in the lab for you to utilize during the setup of your experiments this week. You should make sure to use these sterile shields to keep contaminants from entering your plates through the building's air handling system. When you arrive at your shield, it will be important that you sterilize the surfaces inside your shield before you begin your experiment. We have provided ethanol wipes at each shield for this purpose. Use the ethanol wipes to wipe the inside surfaces of the shield, including the sides, back, and top, as well as the bench top inside the shield. When you are finished, use an ethanol wipe to clean your gloves. Now that you have a clean surface, you will want to make sure that you complete your experiments following some guidelines that will help keep your workspace clean and minimize contamination. When you put pipette tips onto your micropipettes, keep your pipette vertical and make sure not to touch the pipette tip to any surfaces. Make sure that you use a new pipette tip for each step of your experiment so you aren't providing any cross-contamination of your samples. Keep your Eppendorf tubes closed when you aren't using them. Use a new sterile loop for each of your plates so you do not cross-contaminate between samples. If you spill something, clean it up and re-sterilize your workspace. Do not remove any equipment or supplies from the sterile shield. One of the steps in the DNA experiment where we often observe a problem with potential contamination is when students plate their plus plasmid and minus plasmid samples onto Petri plates at the end of the experiment. When you complete this step, make sure that you use a new pipette tip for each solution, one for the plus plasmid solution and one for the minus plasmid solution. Make sure that you use a new sterile loop when you spread the sample on each of your Petri plates. Do not take the lid completely off the Petri dish. Instead, simply tilt it enough so that you can reach the sterile loop inside to spread your sample back and forth across the surface of the agar in the plate. Then, turn the Petri dish 90 degrees and spread the sample back and forth across the surface again. When you are finished with the DNA experiment and you move on to setting up your plant hormone project, you should follow the same sterile technique protocols. Re-sterilize the inside of your shield so you start with a clean surface. Do not open the Eppendorf tubes containing your seeds until you need to add solution, and then close them again when you are done. Use a new sterile loop to spread your seed solution across the surface of each of your Petri plates. The hormone solutions and the sterile water provided to you in the lab this week have been sterilized before they were placed into the repeating pipette dispensers. However, once those dispensers are put into the classrooms, we can no longer control their cleanliness. We are depending on our students to help us keep them clean and sterile so that everyone who uses them will get good results. This means that you should not touch the tubes leading out of the dispenser that deliver the solution. Never open the dispenser to add solution. Leave this to the course teaching staff. Never adjust the volume settings on the repeating pipette dispenser. If you need a different volume, dispense the solution into one of the sterile tubes provided and use a micropipette to get the volume that you need. If you dispense too much solution, dispose of the extra. Do not try to put it back into the dispenser. If you think you may have accidentally contaminated a solution, bring it to your TA's attention so it can get replaced. Once you use the Eppendorf tubes and the sterile tubes for your experiment, please dispose of them in the appropriate location. Do not put them back for other students to reuse. If you have questions about how to set up your plant hormone plates, you should go back and re-watch the Plant Hormone Project video for more details. If you are careful and do a good job of being mindful while you complete these activities, you should be successful in avoiding contamination of your experimental plates. Each member of your group should keep sterile technique in mind and watch for potential mistakes. Don't be afraid to speak up if you see one of your group members make a mistake. Simply correct the mistake and move on to the next step. Don't be afraid to ask your TA for assistance if you need it.